can be a real pain. It's not as easy as coomassie, but smaller amounts of protein you'll be able to see. We're talking sub-nanogram level of sensitivity you can get with a silver stain when you're staining like an SDS page stain. But as you can see by all of these bottles, it's kind of complicated and there's a lot of steps, and you have to be really careful because you can actually, how it works is you're getting silver to like plate your proteins in your gel, and silver can also plate other things, so you need to make sure everything's super duper duper clean or you're gonna get like silver where you don't want it. So we can control the reaction conditions by controlling the pH and various carrier molecules and stuff to control when the silver is actually going to plate out, so hopefully on our protein. And so this can be really useful if you need a really sensitive stain. So let's talk about how it works.
So I'm going to talk about silver staining in the context of staining SDS page gels. So we've separated proteins by size in these gels, much more on that in other posts. And now we want to see where the protein is. And how we're going to do this is by sending in silver ions. So silver in this ionic, this charged state. And it's going to be soluble in this form, and it's going to be dissolved in water, and we're not going to be able to see it. And then what's going to happen is that we're going to let it bind to our protein, and then we're going to reduce those invisible ions to visible solid silver. So we'll talk more about what this means by this reduction, but this is a redox reaction. So oxidation is loss of electrons, reduction is gain of electrons. When you have this reaction happen, the silver ions are going to go from this charged state, from this oxidized state to this reduced state. This is neutral. Water doesn't want to hang out with it, and it's going to precipitate out as this solid silver metal. It's going to like plate our proteins. So this is actually the concept behind like old school photography, like development of film. And this method can also be used to stain other types of gels like agarose gels. It can even be used in like histology, people like staining nerves and stuff in brain tissues. So a lot of different uses. I'm going to be talking about it in the context of staining an SDS page gel. And I'm also, another note is that there are a lot of different methods for silver staining, and the method that I'm going to be talking about is the one that our lab uses, which is this Bio-Rad system, which is uses this silver diamine staining method. But a lot of the concepts that we're going to talk about are the same no matter what the technique you use. And so hopefully this will be helpful even if you're not using that same kit. And again, I don't work for Bio-Rad or anything. That's just the kit that I've used and that our lab has. But let's get back and talk more about how this is actually going to work. So as I mentioned, silver staining is very, very sensitive. So it's about 50-ish times more sensitive than the typical like coomassie-based stains. But it's not without cost. And so as you can see by all the smudges and stuff on the gel, by all of this dark blackness on this gel, it can sometimes be too sensitive. So what can happen is you can get artifacts. So you can have it staining things that aren't your protein, and you can also overstain it. Unlike the coomassie, where basically you can wash out the stain that's not bound to proteins, when you're doing a silver stain, the reaction is actually developing inside of the gel. And if you let it go too long, it's going to start just staining everywhere and you end up with a totally black gel. So while you're there watching your reaction, watching these bands develop, you're trying to figure out, okay, what time do I actually want to stop this so that I get the stain, so I see the bands that I want without just staining all of the background. And so how do we start and stop the reaction? How we're going to do that is by controlling the pH. So this reaction is relying on hydroxide ions. And going back to acid-base chemistry, which I talk about more in other posts, this is you'll have more of these under basic conditions, whereas under acidic conditions, you're going to have more protons or more hydronium ions, so those protons linked up to a water molecule. And so pH is a measure of the free proton availability, but it's on a inverse log. So the higher the pH, the lower the concentration of protons and the higher the concentration of the OH. And so the higher the pH that we're talking about basic conditions, and if we're talking about a low pH, we're talking about acidic conditions. And we're going to take advantage of both of these in the silver stain. We're going to use acidic conditions when we're getting everything set up to prevent this reaction from happening too soon. Then when we want things to happen, we are going to add a base. We're going to make a basic conditions, and this is going to allow the reaction to occur. And then we're going to add an acid again to get it to stop before our gel is just stained all over. So the acid and base is going to let us control the reaction. And then this reaction itself is going to rely on this oxidation and reduction reaction. So remember, oil rig, oxidation is loss of electrons or electron density, and reduction is gain of electrons or electron density. And so what's happening when we have this reaction is that this is the oxidized form of the silver, and it's going to be dissolved and invisible. As we'll talk about, it's also going to be complexed with other like carrier molecules and ions. It's going to help shield it before it finds the protein. Then what's going to happen is it's going to get reduced to this solid visible form of silver metal. But when something gets reduced, something else has to be getting oxidized. And the thing that we're going to get oxidized is going to be formaldehyde, which is going to get oxidized into formate. So here you can see oxidation is often, but not always, accompanied by the gain of bonds to oxygen. And so you can see here that we have formaldehyde is gaining the bond to this oxygen. And because we're under these basic conditions, this is going to be formate instead of formic acid, where you have the H here. But then what we're actually going to do is we're going to add acid later, and then when we stop the reaction. But this is the key reaction. So you have the silver getting reduced, the formaldehyde getting oxidized. Just a note about this reaction too is that a similar reaction is used in a chemistry, like an organic, something you might have done in like organic chemistry. If you're doing one of those unknowns tests, unknowns labs, there's like this Tollens test, which tests for the presence of a ketone versus an aldehyde. And so with formaldehyde, we have an aldehyde, which means that we have a carbonyl, so a C double bonded to an O bonded to a hydrogen. When you have a ketone, you have a carbon on both sides of that carbonyl group. And this is not going to give you it's not going to serve as a reducing agent in this reaction. And so you are not going to get silver plating with a ketone, but you would with an aldehyde. And so in this Tollens test lab, you can actually test and see, okay, well, if I add these reagents to this unknown compound, do I get silver plating, which would tell me that I had an aldehyde as opposed to a ketone if I knew that I had one of those. Okay, so let's go over the basics of the silver stain. So no matter what the method is, typically the steps are going to go something like this. First is the fixation. In this step, you want to get your protein stuck in the gel. Commonly, this is done with a solution that has like acetic acid and methanol. These are going to precipitate out your proteins, get them to like be unhappy and clumpy, and this is going to keep them from like diffusing out of your gel. So they're kind of trapped in the gel, but they can still kind of wander around, and you don't want them to be wandering around and wandering out of your gel and stuff. We need the silver to find them in place, and so we need to keep them in place, and how we can do this is with this fixative. In the Bio-Rad kit that I use, it has this like fixative enhancer concentrate. This is like a glycerol solution, and it's supposed to somehow like enhance things and also prevent the shrinkage of the gel that can come when you're doing this fixation step. Then you're going to wash it. So you want to wash out the fixative and fill the gel with super, super pure water. Throughout this process, it's important that you use really, 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 really pure water. So you need to be using like this milli-Q water. Basically, if you have any impurities, then if you have like ions and stuff, because our solution is going to have all these ions, you can actually get them like precipitating out the silver, which can then cause problems and hide the silver from actually getting to your protein. And if you, so like deionized water can remove those ions, but there can still be little particulate stuff. And if the silver binds to the particulate stuff, then it can plate out on the particulate stuff instead of on your protein. And so you background problems. So what we do is with this milliQ, these extra filters that are going to give us this really pure water. And so we want to use this really, really pure water and wash out our gel. We typically do like 2 washes because this is relying on diffusion. And so diffusion is going to get things to like flow out of the gel until there's like into the gel until there's like an equal concentration of things inside and outside. And then because you're sending stuff out, now that stuff out is going to get in equilibration. And so in order to get it like all out, you can never really get it all out, but in order to get it mostly out, you want to then replace the water and do another wash and to get everything out before we actually get this reaction going. I'll talk more about the actual how we do this staining step. Some of the methods combine these steps, like the one I do, but some of these do these separately. There are 2 main things. You need to send in the silver ions and get them to bind to the protein. And then you need to actually get those to reduce to be the solid silver. So we get our protein stuck, we wash out other stuff, we get the silver to bind the protein, and then we turn the silver into a solid. And then we need to stop it. So we need to stop this reaction before you overstain and before the background is too high. We'll get into, there are a number of different methods, different strategies that people use in order to do like sensitization or an enhancement to increase the signal to noise. And this might be as its own step. Sometimes there's like after this wash step, you have like an this sensitization step, or it might be combined with other steps depending on your method. And often there's like a combination of different methods that are used in order to try to enhance things. Okay, so in this kit that I'm using, and remember that this, like a lot of techniques out there, you don't need a kit, and they'll be similar to like this. And so I looked into the various components that are actually in these different reagents to try to like make this so that, well, first that I knew what the heck was going on, because I don't like black box type things. I like knowing what's going on. And also then hopefully that this can help you understand things if you're using a different method that you don't have a kit or that sort of thing. Okay, so the fixative. We already talked about the fixative. That's going to be like our methanol, our acetic acid, and then like the glycerol in this case. Sometimes there might be other sensitization agents. And now our stain. So with this stain, it actually, the 4 of these bottles are all contributing to the stain. And so we need to mix them in a specific order, because it's really, really important to make sure that the formaldehyde is going into acidic condition, so it's not going to react too soon. So the 4 components that we're going to have are going to be the silver complex solution, which is going to be silver nitrate and ammonium nitrate. We'll talk more about why we have these 2 things. And then we have this reduction moderator solution, which is going to be tungsticilic acid. And again, we'll talk more about what these do, but these are going to serve as carriers to kind of carry the silver into the gel and prevent it from reducing too soon. Then we have this image development reagent, which is going to be our formaldehyde, and that's going to be our reducing agent that's actually going to get things, get the reaction to happen, get our silver to be reduced. And then we're going to add this development accelerator solution, the sodium carbonate. So this is going to be make our conditions basic to get this reaction to occur. And this is why we need to have this all set up before we add this. So it's important that you're making this stain fresh, like right before you're doing the reaction. So like when you're doing that wash step is a really great time to be preparing this. They all come as solutions except for the development accelerator solution for our sodium carbonate. And so this, it's only good for a little bit. So if you're going to be if you're not going to use it all within, I think it's like a month or something, then you want to just like weigh out the amount of the powder that they give you to make the amount that you need, so you don't waste it all. But you want to be adding this, like especially this one, you add it right before you're ready for your stain. So you're going to mix these, then add this, and then add this right before you're ready to pour it onto your stain, because we don't want any of the side reactions to be happening before like we're actually even like on our protein. So we want to get the best possible chance to have the low background. And then we want to stop it, and then we can do that with this like 5% acetic acid. So we're going to be tightly controlling the pH. And this is why we need to do things in the specific order and make sure that we're setting things up in these low pH conditions, so in these acidic conditions. So under those acidic conditions, we're not going to have enough of the hydroxide ions for this reaction to occur. And then when we add our base, we're going to increase the concentration of the hydroxide ions to get this reaction going. And this reaction is going to go really quickly because the silver plating is autocatalytic. This means that like once it starts, it's going to nucleate further deposition. So you can think of the first like silver to get reduced as kind of being a trendsetter, and once it sets that trend, then a lot of other silvers are going to hang out too. It's more just like it's kind of making a surface that the silver can then latch onto, so it makes it easier for the other silver molecules to plate out. And so this is why it's really, really important that we want this to happen on our protein and not on our background, which is why we're taking so much care in order to tightly control when and where the silver is actually going to be reduced, where that trend-setting reaction is going to occur. One of the ways that we do this is by actually like hiding the silver ions. So the method that I'm using uses this diamine stain. And so so 2 amine, so like these ammonia with hydrogens. And so this is ammonia. This is an ammonium ion, and so it's in this positively charged state. And so when we have ammonium nitrate and when we have silver nitrate, we can write them like this, but when we dissolve them in water, they're going to dissociate into ions, so charged particles. And we're going to end up with like our silver ions, we're going to have these nitrate ions and ammonium ions. So if you have acidic conditions, under acidic conditions, there are a lot of protons. And so this is going to be positively charged. This ammonium is going to be positively charged. And so it's not going to want to hang out with the silver. But later, when we have the basic conditions, this is going to deprotonate to give us a neutral ammonium. And then this will hang out with silver in this diamine complex. So we have 2 ammonium per silver. And they're just kind of like hanging out. They're not like tightly bonded, but silver has this big electron cloud, and so it can form these sort of complexes with other molecules. And so you get this diamine complex, and this is going to help like lower the free Ag plus concentrations. So if we just had ammonium nitrate, so if we just had silver nitrate, we would have these silver and these nitrate ions hanging around. And we would have a lot of free silver ions, which could then be susceptible to accidentally reducing, not accidentally, but just reducing where we don't want them to happen. And so we want to hide the free silver concentrations to lower the background. And so this diamine stain is the one way to do that. The other way we're going to do that is with this other carrier molecule, TSA, so tungstosilicic acid, aka silicotungstic acid. So the key thing is you have the silicon with these oxygens. So under acidic conditions, these are going to be protonated, but under basic conditions, they'll deprotonate. You can get this negatively charged compound that can then hang out with the silver diamine complex. So you get this overall like complex, and this is serving as like this carrier molecule. And so this can help bring the silver into the gel, while shielding it from premature reaction. And then once the silver finds the protein, it likes the protein better, and so it can ditch those guys and bind to the protein. As to how the silver is binding to the protein, there's a variety of different ways. So these silver ions are positively charged. So the most obvious thing is that they could bind to negatively charged amino acid side chains, such as glutamate and aspartate, because you have this opposite charge attraction. You can also have partly positive regions of proteins. So because the electrons aren't shared fairly, you get this like polarity, and you can have interactions like that. You can also have like temporary or induced dipoles. So partial charge separate, you get these partial charges that's kind of just like happening randomly, or because the silver ion is nearby and it can kind of shift things. So there's a lot of different ways that silver combines, which means that it can actually have better. Sometimes there are proteins that won't stain well with a coomassie stain, because coomassie stain is going to be biased more because it only it prefers to bind certain amino acids. And so although the silver stain has some preferences, if your protein doesn't stain well with coomassie, it might stain better with the silver stain. And in addition, the silver stain is going to be a lot more sensitive, but remember, sometimes too sensitive. So speaking of too sensitive, there are things that you can do in order to try to increase the signal to noise ratio. These are sometimes called like sensitization methods or enhancement methods. And so they're all trying to improve the signal to noise, but they take a bunch of different strategies in order to do this. One is to increase your signal. So you can do this by making the proteins more attractive to the silver. So as I said, silver can kind of bind to a lot of proteins in a lot of different ways. And it can have better binding than coomassie dye. But if you want to really increase it binding to protein as opposed to other things, because you can imagine if it can bind to protein in a bunch of ways, it can bind to other stuff in a bunch of ways. And so you can pre-bind other compounds to the protein that also bind to silver. So there are a bunch of different compounds that are tried for this. This is also going to allow for amplification because it's increasing the number of silver binding sites on your protein. You can also decrease the noise. So you could reduce the background staining. How you can do this, so thoroughly wash out those electrophoresis buffers. You can add chemicals to sequester the unbound silver ion so it can't react. And so this is what we saw with like our carrier molecules. And you can control the speed. So you can either speed things up or slow things down. So you can speed up the start to promote the initial reduction if you pre-bind the reducing agents, such as glutaraldehyde, to the protein. So they're right there and ready to initiate the reduction. So basically, when you have, before we were talking about pre-binding the things that bind silver, here we're talking about pre-binding our reducing agent, so that once the silver gets there, things are already ready to go. Another thing that you can do is and then another thing about this is that, so like formaldehyde, it can form these polymers, so it can like link up into these chains. And so that's another reason why it's really important that we're setting up the reaction, preparing our stain in that specific order in order to prevent that. And so we don't want these like side reactions taking place. Another way to control the speed is to do kind of go the other direction, but at a different step. So we want to slow down the continuation of the plating. So here we're trying to speed up that initial trend setting reaction. Here we're trying to slow down all those like wannabes that are following along. So we can slow down the continuation of silver plating. And this is going to give the protein time to loosen its grip on the bound silver so it can be reduced on site. So when the silver is bound to the protein, it's hard for that silver to get reduced. So it has to kind of like loosen up the grip a little in order for the silver to actually be reduced on the side of the protein. And if you slow down the continuation of the silver plating, such as by using mild or reducing conditions, this can help give this reaction time to occur. So it's kind of you have to kind of get a sense as to when to actually stop it before your reaction is too far. but you can stop it just by like adding acetic acid. When you add this acetic acid, now you're going to be taking away those hydroxide ions. So now they're going to be protonated and form water. You're going to be protonating that ammonia. You're going to be protonating your TSA. So basically, you're making the conditions that we were setting things up in, but the conditions that we then took away so the reaction could happen. Now we're bringing back those conditions that are going to keep the reaction from actually happening. So as I mentioned, the silver stain can be very, very sensitive. And it can also, as you can see from all this background staining that we've been talking about ways to avoid, this staining can also take place on things like your gel box. And so you want to try to avoid that from happening by thoroughly washing everything beforehand. So you want to try to avoid that in the first place, but it doesn't always work perfectly. And so you need to make sure that afterwards, you're going to be cleaning out your gel box really, really well. I like to keep separate, separate containers and stuff that I use just for silver staining, as opposed to the containers that I use for other, for like coomassie and stuff, so that I keep the silver staining stuff really, really clean. And how I can clean it, so you can see this like silver deposited, you can actually add nitric acid. And then this is going to get our silver, that is this solid reduced silver. This is actually going to turn it into that silver nitrate ionic compound. And so this is going to be soluble. And then this is going to, you can then dispose of with like the hazard, whatever the hazardous waste condition protocol is where you are located. And so this is the basics of silver staining. And so I haven't had that many instances where I've actually needed to use it. But for some, if you're doing like a pull down or something and you're going to do mass spec and you want to see if there are protein bands actually there, other times you might want to be seeing if there are degradation products, and when, if so, when do those degradation products first start appearing? You can, because it's so sensitive. If you have a really dilute, for some reason you have a really dilute sample and you want to see what's in it without having to concentrate it. If your protein doesn't bind well with coomassie, as we've been talking about. Another time you might want to use it is if you're trying to look at both a protein and DNA or RNA. So as I mentioned, you can also use this to stain for nucleic acids. And in fact, the protocol that the Bio-Rad kit is based on, they send you to this citation that's actually looking at DNA in agarose gels. And so, yeah, it's pretty cool that because silver is able to bind into so many things, it's also able to then show us their presence in a gel. But also another good reminder that you need to be careful that you make sure that the silver is only binding to stuff that you want it to bind to. And so we need to make everything else really, really, really boring, washing everything out really well. And so I've never actually used it for nucleic acids, but apparently the RNA is going to like stain browner, and then the protein is going to stain blacker, and you can actually even tell the two apart if you have a mixture of them. So pretty cool stuff. Maybe I should try that someday. So there are a lot of different reasons why you might want to use a silver stain, but as you've seen, it's kind of a pain.